We just finished discussing RNA splicing. So now you know how humans and other animals can make many more proteins than we have genes. So pretty cool idea. But let's go back to the idea of transcription and translation. In many ways, the nucleus controls what each cell in your body does. And the way the nucleus controls each cell in the body is by dictating which proteins the cell is making. So, for example, we have got all kinds of instructions here inside of the nucleus of this cell, and all of those instructions could theoretically allow this cell to become a beautiful eyeball cell, or a beautiful spleen cell, or a beautiful stomach cell. All of those instructions are right there in the form of your DNA. The DNA in each cell of the body is almost identical. There are exceptions, okay? But this particular cell is going to become a stomach cell. Why? Because the nucleus has decided. Okay, well, how does the nucleus make that happen? The nucleus will only transcribe genes that will build proteins that will allow this particular cell to become a great stomach cell. So when the nucleus decides I'm going to be a stomach cell, then it will transcribe and splice messenger RNA, and that messenger RNA will go out here to Mr. Ribosome. That's a ribosome. And the ribosome will simply build whatever protein it is given the messenger RNA for. Has the ribosome decided what to make? No. The nucleus decided what the ribosome should make, sent out just those instructions, spliced, ready to go, and now the ribosome will build that particular protein. Remember that ribosomes are the machines that make proteins, and they can be free out in the cytoplasm or they can be attached to the rough endoplasmic reticulum. Replication is going to happen only once in the life of a cell, and DNA does that, okay? Transcription, the DNA will transcribe messenger RNA. So at the beginning of transcription, you've got just basically a gene of DNA, and then you will end up with messenger RNA that's going to leave the nucleus. The DNA stays in the nucleus where it's safe. Translation is going to happen when a ribosome uses that messenger RNA copy of a gene to build the protein. I'm sorry, this image didn't turn out right, but here we have got an entire cell, and out here, these little purpley dots, those are ribosomes. And the ribosomes could be free out here in the cytoplasm or attached to the rough endoplasmic reticulum. But when the nucleus decides what needs to be built, then the nucleus sends out messenger RNA that the ribosome will turn into a protein. DNA is not directly involved in translation, but DNA is involved in transcription. So let's talk a little more about translation. In order to get translation, we already know that you needed 3 different types of RNA. You need messenger RNA, transfer RNA, and ribosomal RNA. You've learned more than you ever wanted to know about messenger RNA, how it gets spliced and all that stuff. The little ribosomes, those machines, they are made out of ribosomal RNA and protein. Those little machines are going to actually build the protein by connecting amino acids with peptide bonds. So what is this transfer RNA? The transfer RNA is going to escort the amino acids one at a time to the ribosome so that the ribosome can assemble them. Let's look at transfer RNA. Transfer RNA, every transfer RNA carries a specific amino acid. This is what a transfer RNA basically looks like. It's kind of a weird 3-leaf clover shape. At one end of the transfer RNA is carrying an amino acid, not just any amino acid. Every transfer RNA is carrying the amino acid that this end, its anticodon, will recognize the codon for that amino acid. You know, that was very clearly stated, but it probably didn't make a whole lot of sense. Okay, for example, if this is the amino acid that's carrying leucine, then this anticodon would be the complement for the codon in the messenger RNA that says put a leucine here. Okay. Now, transfer RNAs are the only RNAs that have anticodons. Why are they called anticodons and not codons? Well, codons are not just any 3 amino acid sequence of DNA or RNA, right? It's not that. Codons are 3-letter code words for a specific amino acid. So the sequences of nucleotides that are found in, for example, those introns that we talked about, those are sequences of nucleotides, but we would not call them codons because they are not part of a little code message for a sequence of amino acids. This one is not a codon because it's not part of a message. It's also not just a bunch of nucleotides. It gets its own name, anticodon, because it will specifically bind to the codon in the messenger RNA at the right time. Right? So DNA transcription is going to take the DNA blueprint and make an RNA version called messenger RNA. RNA splicing is going to remove the introns and maybe rearrange the exons, and then you will end up with mature RNA. That mature RNA is going to leave the nucleus through a nuclear pole, and then in translation, that messenger RNA is used as a template. It now is a series of codons, and the ribosome will read it one codon at a time and bring the right transfer RNA, the one whose anticodon matches that codon, to the growing protein chain because it is bringing the right amino acid to the growing protein chain. Transfer RNAs bring the amino acids to the ribosomes so they can be joined into a new protein. Let's look at the image in your textbook for how this happens. All right, so I hope you can see this well. Let's start with number 1. Step number 1, we have got focus mouse, hide cursor, highlight clicks. I don't know, I feel like I need more. Okay, so we have got right here, this is the messenger RNA, and it is a series of codons. Here's the one part of the ribosome. Ribosomes come in 2 pieces, 2 subunits. Here's the first subunit, and it is going to bind when a transfer RNA, that's depicted by this little blue curly Q thing, binds its anticodon to the start codon. Start codons in animals are AUG. And the transfer RNA that has the anticodon for AUG carries the amino acid methionine. I don't think that's on this exam, but it's true, okay? Now, once this happens, then the next subunit binds, and now we have got a completed ribosome. And we're ready for the next transfer RNA, which you can see here. The next transfer RNA needs to have an anticodon that binds to, let me make this a little bit bigger. I made it smaller so that we could see the whole thing at once, but I can see that that was a bit of a mistake. I don't care if you've updated things. Okay, so here we have got our completed ribosome, and here you can see the next codon is UGC. Does the ribosome know what UGC means? No, the ribosome's not very smart. But the ribosome does know that it needs to grab a transfer RNA whose anticodon is the complement for UGC. So what would the complement be? ACG. Here's happens to be ACG. ACG carries an amino acid. Which one does it carry? Okay, they don't say here. And also, the ribosome doesn't care. The ribosome knows that if this is the anticodon for that, then this transfer RNA is carrying the right amino acid. So the ribosome will allow this transfer RNA to come in, and its anticodon will bind to that codon. Now it's time for the ribosome to do some work. It is going to attach the first amino acid to the second amino acid. And at that point, we have actually got a new peptide bond, and we have got two amino acids stuck together. What's going to happen to this transfer RNA? This transfer RNA is no longer carrying an amino acid, so this transfer RNA is ready to leave. The ribosome will move down one codon, and now the first transfer RNA can leave. Maybe it'll go pick up a methionine and come back later, right? But in the meantime, this ribosome is ready for the next transfer RNA. Now, the next codon is CUG, so this ribosome is looking for a transfer RNA that has an anticodon that is GAC. When the GAC gets brought in here, then the GAC transfer, the transfer RNA that has the anticodon GAC, when it gets brought in here, it is carrying the proper amino acid. The ribosome will attach these two guys to the new amino acid, and now this transfer RNA will be allowed to leave, right? So one at a time, the ribosome is going to read the messenger RNA, one codon at a time, bringing in the right amino acid, not because it knows what the right amino acid is, but because it brings in the right transfer RNA. And then the ribosome will be responsible for creating peptide bonds between the amino acid chain that's already been built and the new amino acid that just got brought in. So you can see that one little nucleotide at a time, one amino acid at a time, the protein is going to be built until you finally have the completed protein. Great. Alrighty, we're going to pick up here at the beginning of the next video.